Hello all and welcome to Talk GCSE. Today we're going to talk about monoclonal antibodies. Now this topic links to things like immunity, biology in medication and genetic engineering. Now by the end of the video you should be able to explain what monoclonal antibodies are, describe how they are made, explain how they're used in medication, diagnosis, pregnancy tests, and evaluate their advantages and disadvantages. So let's start off with antibodies. Now, an antibody is a protein that is made by white blood cells called lymphocytes. Each antibody has a specific shape that allows it to bind to one specific antigen. Now, an antigen is a molecule on the surface of the pathogen, such as bacterium or virus. Think of antibodies like locks and antigen like keys. When an antibody binds to an antigen, it can neutralize toxins, it can cause pathogens to clump together, it can mark pathogens so they can be destroyed by white blood cells. Now we go on to the word monoclonal. Mono actually equates to one, and clonal is from a single clone of cells. So think about monoclonal antibodies. They are identical antibodies, they're produced from one type of cell, and they all bind to the same antigen. This makes them very specific and, in turn, very useful. Now, if you're doing higher tier, this is the content that you also need to learn. How are monoclonal antibodies made? Now, there are quite a few steps here, so we want to be fully concentrated here. And if you're writing these down, you may want to stop the video, write down your points, and then restart again. Step number 1, injecting an antigen. So, a mouse is injected with a specific antigen. The mouse's immune system responds by producing lymphocytes that make antibodies against that antigen. Now I want you to think an antigen, think of it like a specific barcode of the pathogen or the bacteria. Step 2 is collecting lymphocytes. Now the lymphocytes are removed from the mouse's spleen. However, lymphocytes don't live very long and they can't divide forever. Step number 3 is combining them with tumour cells. Now the lymphocytes are fused with tumour cells. Tumour cells, they divide rapidly and they live for a long time. The fusion are called hybridoma. So the fusion cells are called hybridoma cells. So when we say the word hybridoma, I want you to think antibody producing and fast dividing. Step number 4 is cloning the hybridoma. Now hybridoma cells are cloned to make many identical cells. Each cell produces identical monoclonal antibodies. Step number 5 is collecting those antibodies. Now these antibodies are extracted, purified, used in medicine and testing. Now we need to understand the uses of monoclonal antibodies. They can be used in pregnancy tests. So monoclonal antibodies bind to the hormone HCG in urine. If HCG is present, antibodies, they bind, a coloured line appears and test is positive. This works because monoclonal antibodies are very specific. We can also use this to diagnose a disease. They can be used to detect things like cancer cells, specific hormones, pathogens. Antibodies are often attached to fluorescent dye. If the antigen is present, the antibody binds and the area glows and doctors can see where the disease is. And thereafter, they're used to treating disease, targeted drugs. So monoclonal antibodies can be attached to drugs, attached to radioactive substances. They bind only to target cells, for example, cancer cells. What that means is less damage to healthy cells and more targeted treatments. They do have their list of disadvantages and risks. Monoclonal antibodies are very useful. but there are problems. They can cause side effects such as things like fever, vomiting, low blood pressure. They are expensive to produce. And some people are concerned about using mice, so there's ethical issues that arise. Now, in terms of exam, so let's just summarize all of this. What you must remember is that monoclonal antibodies are identical antibodies. They bind to one specific antigen. They're made using hybridoma cells. They're used in things like diagnosis, treatments, pregnancy tests, and they do have the fair share of advantages and disadvantages. So if you can explain this, explain the process, and evaluate their use, you are exam ready. And if you found this video useful, don't forget to follow Talk GCSE. Thank you.